Hello, everyone. Welcome. I think we're going to start promptly with the introduction just to avoid eating up time for the talks. And I'll just get right on it. So let me just see if I can rearrange my desktop a little bit. Okay. So welcome again to the MultiHEP workshop. This is intent for multidisciplinary development for high energy physics. This is a SNOMAS pre-meeting, which is organized by the cross-cutting group of the instrumentation frontier, IF9. And the focus is on high energy physics developments that make use of expertise from outside high energy physics through collaborative efforts. As you know, there's lots of examples of high energy physics technology applied to other areas. And these are more familiar, they get more airtime because they're good for outreach and also for funding agency justifications and so on. So the point here was to look at the other side of the coin and see how we're doing on bringing in other technologies to carry out high energy physics experiments. And the workshop will provide input to a SNOMAS instrumentation frontier report. So we'll collect abstracts or summaries from the presenters and notes from the panels and share that. So can everyone hear me okay? I'm assuming this is all going well, but I haven't heard anything. I see lots of people connected.
I can hear you.
Yes, Maurice.
Yes? No?
Yeah, we can hear you, Maurice.
This is all going well, but I haven't heard. I see lots of people connected.
I can hear you.
Okay,
Yes,
thank
Maurice.
you. Yes. Okay, I'll continue then. I'm on slide 2.
Yeah, we can hear you, Maurice.
Oh, okay. To check. So the workshop is hosted by the Berkeley Experimental Particle Physics Center, BEP. Okay, I'll continue then. I'm on slide 2. Oh, I need to, sorry. Let me, there's
Yeah, we can hear you, Maurice.
okay. To check. So the workshop is hosted by the technical troubles. Berkeley Experimental Particle Physics Center, BEP. Okay, I'll continue then. I'm on slide 2. There. Okay, sorry about that. There was I had a YouTube window open, so I'll get to that in a minute. And it's also associated with the ICFA Instrumentation and Innovation and Development Panel and the local organizing committees from Berkeley, because we're virtually hosting it in Berkeley. And then there's a scientific organizing committee with the people listed here. So the timetable, which you can see on Indico, so we have 3 half days. We're skipping over Wednesday to observe Veterans Day. And I hope you will connect all 3 days. The times are the same all 3 days. So there's starting at 8 a.m. Pacific. So today we have some talks and then a panel. Tomorrow, 2 panels, no talks. There'll be short introduction. And then on Wednesday, like I said, we're taking a break. And on Thursday, we have again talks followed by a panel. So there's the usual etiquette for these remote meetings. So please follow the SNOMAS deep principles and guidelines, which you can find there. The talks that we have featured are not intended to be a comprehensive survey of all activities, all multidisciplinary activities, but the idea is to provide moderation for the panel discussions. We are going to record the talks on panel, so I am not recording this. I'll start recording for the first talk, and then we'll distribute the links so that you can watch later. Also, you will notice if you go to the agenda now, there's YouTube links that you can follow through live streaming on YouTube if you prefer, and this allows you, of course, to go back and forth on the video, so you can replay things as often as you like, and as long as you're not talking, then it's just it's the same thing as watching on Zoom. We have limited times for questions after the talk, so please use the raise hand feature for questions, but most importantly, just submit questions to the form on the agenda that says, submit a question on the timetable, and this is what we prefer before the panel start. Once the panel start, though, we're not going to be updating the form, or looking at the form updates. So just use the chat window during the panel, and hopefully you'll provide further feedback even after the meeting. Like I said, this is all going to result in a report. So you can organize or use again those forms that are on the agenda. So that's it for the introduction. And I hope you enjoy the workshop. I'm going to pass it on now to the convener of the first session, who's Petra, who I have to make host.
Hi, good morning. Yes, so the first session at this workshop is focusing on biology, material science, and chemistry as other disciplines and their input into HEP. And our first speaker is Ben Jones from University of Texas in Arlington, talking about the chemistry for barium tagging. And Chris, you have 30 minutes. I'll give you a warning when there are 10 minutes left. So there should be some time left for questions as well, if you can.
Okay. Do you see my screen?
Yes.
Do you see my mouse as well, or just the screen?
Yeah, I see the mouse.
Great. Okay, so thank you for inviting me to this workshop. Very excited to be here. I'm presenting work from a wider group of people than just myself. In particular, this is an interdisciplinary collaboration between the group of myself and Dave Nygren at UT Arlington and Frank Foss, who's our collaborator in the organic chemistry department at UTA. So I sort of anticipated a wide array of people from different backgrounds at this workshop, and so I am going to give just a few slides introducing the problem of neutrinoless double beta decay before getting into the chemistry part. But essentially, it's one of the top priorities in neutrino physics today is to understand whether the neutrino is a Majorana particle. If the neutrino is a Majorana particle, then at least at sufficiently high energies, that means it's a kind of particle where when it's spinning one way, it behaves like matter, and when it's spinning the other way, it behaves like antimatter. If it is one, it's the only one that there is in the standard model. It's particularly interesting to ask the question about whether the neutrino is a Majorana particle, because it wouldn't just be, a measurement of something unmeasured. That's interesting in itself. But it would tell us 6 really fundamental things about nature before breakfast. If we were to find that the neutrino is Majorana, we would immediately know that lepton number is not conserved. We would know that there are massive fermions that exist that are neither matter or antimatter, but something else. The standard model then with this Majorana mass term is a non-renormalizable theory. So we would know that the standard model is definitely a low energy effective theory, one way or another. We'd know that there are other mechanisms for generating mass in nature beyond the Higgs mechanism, and Higgs may be involved here, but there would have to be another ingredient to the mass generation to create the Majorana mass term. We would also, just like once upon a time, the Fermi scale gave us the masses of the W and Z. This would tell us the neutrino mass probably tells us the next scale, although there's a probably caveat here. You can always build a model where it doesn't. And then finally, Majorana neutrinos are a prediction of leptogenesis, and that's one of the most compelling theories for generating the observed matter-antimatter asymmetry of the universe, assuming you have enough CP violation, and that needs to be addressed by other experiments. So it's well motivated to look for this, and it tells us several very, very important things if we find it. The way we look for Majorana neutrinos is via searches for this process neutrinoless double beta decay. So double beta decay is a regular kind of radioactive process, which is predicted to have a finite rate within the standard model, and it's been observed with rates consistent with expectations in multiple isotopes. A double beta decay is a decay whereby a nucleus, because of the nuclear pairing force, a particular isotope is energetically allowed to decay with 2 beta emissions at once, but energetically forbidden to decay through a single beta decay. The typical final state of an allowed double beta decay then is 2 electrons and 2 antineutrinos, or 2 positrons and 2 neutrinos, depending on if it's double beta plus or double beta minus. This has a half-life 10 to the 19 to 10 to the 21 years. The more interesting kind of double beta decay for neutrino physics is neutrino-less double beta decay. This is a process whereby no neutrinos emerge, only electrons, and this is allowed if and only if the neutrino is a Majorana fermion. And its half-life depends on some product of factors, which include things about nuclear physics, as well as things that we don't yet know about the neutrino masses and mixings. So the rate that this process goes is not presently known, but it appears, based on experimental limits right now, it's at least 1 million times slower than the allowed double beta decay process. And if we see it even once with sufficiently low background, then we would know the neutrino is a Majorana fermion. So in principle, a single event discovery is possible in this field. So the ideal experiment then, we look for these electrons and we measure their energy. And in the case of the neutrinoless double beta decay, all the energy in the decay comes out in the electrons because the nucleus takes almost nothing in the recoil. In the allowed double beta decay, the energy is shared between electrons and neutrinos. And so we see these 2 characteristically different energy spectra, a broad spectrum of energies in the allowed case and a narrow spectrum in the neutrinoless interesting case. And so if we can measure those electrons and measure their energy sufficiently precisely, that lets us tell that we're in the red spectrum and not the blue one here. That requires, in practice, an energy resolution something like 2% full width half max, which has now been achieved in multiple technologies. But then really what we need is no other kinds of backgrounds from radiogenic or cosmogenic activity in this region of interest. And if we can achieve those 2 things, the good enough energy resolution to reject the 2 neutrino mode and the lack of any other backgrounds in the ROI, then you would just watch and wait for some atom in your detector to undergo this process. Of course, that case which is ideal is never what we really have in these experiments. There's always some amount of background, usually from radiogenic activity at the scales that we presently live at. This plot shows you how your sensitivity goes as a function of exposure for various different levels of background. The background free case is the solid line, and here you might cross what we would loosely call the interesting parameter space, although I don't want to get into a deep discussion of what interesting parameter space means necessarily in this talk. But you would get to cross interesting parameter space in 2 to 3 years in a background free experiment, which was 1 ton. If you have background rates which are more like 0.1 count per ton per year in the region of interest, that will take you 15 years. If you have 1 count per region of interest per ton per year, you would take 100 years. And if you have 10 counts of background per ton per year expected, it would take you 1,000 years to cross that piece of parameter space. So really reducing the backgrounds is of critical importance. Now, there's a whole field of neutrinoless double beta decay technologies, each one more beautiful and complex than the last. And they all have different approaches to these problems of reducing the backgrounds. But this plot kind of summarizes how everybody is doing at the present time. So where we need to get to really is somewhere below that dashed line at the bottom to get into the really interesting parameter space. And at the 100 kilogram scale, most programs are between a factor of 10 or 20 to 100 away from crossing this line. So in order to have truly sensitive future double beta decay experiments, some dramatic improvements in background are required. And of course, every collaboration that's on this map has a proposed plan for how they're going to do it and how they're going to get the backgrounds down and get across this line. So it's clearly very challenging, and the experiment that I work with next uses high pressure xenon gas time projection chambers. The idea here is to achieve a very big, uniformly low background double beta decay detector with active background projection, rather than relying on so much on material radio purity and shielding. It's a phased program. I'm not really going to talk about it today because what I'm really here to talk about is barium tagging. So barium tagging is a proposed method of reducing the backgrounds in xenon-based double beta decay experiments to nearly negligible levels. And the idea here is that in one of these double beta decays, you produce not only 2 electrons with a precisely known amount of energy, but you produce a barium ion. And if you can identify that barium ion, that tells you that this activity that you saw was not from a radiogenic event, because a gamma ray scattering in the detector can't make an isotope hop 2 places across the periodic table. So a barium ion detection in coincidence with an energy measurement of 1 or 2% full width half max, depending on how big the experiment is. You need better energy resolution the bigger it is. But a combination of those 2 things would give you an in principle background free signal, enabling a more robust discovery claim and entering new regions of sensitivity. So this is not a new idea. There's been 30 years of exploration about barium tagging in neutrinoless double beta decay. And it's been explored for both liquid and gas detectors. Actually, it's only in the last couple of years that really, really exciting developments have been happening, and this starts to look more plausible. So this is by its nature a widely interdisciplinary topic, and so I'm very happy to talk about it here. It combines elements of nuclear physics, organic chemistry, atomic physics, analytic chemistry, microscopy, gas microphysics, and mass spec. One of the reasons we enjoy working on it so much, actually. To talk about the reason we invoke organic chemistry for gas phase barium tagging, I need to quickly talk about the nature of the barium you're looking for in this decay. So the barium is born in a violent radioactive process, or at least violent on the scale of atomic electron binding energies. And it actually is born in a very highly ionized state, very many pluses, barium plus, plus, plus. But what happens immediately after that decay is that it thermalizes and then it captures electrons from the nearby xenon around it until that's not energetically favorable anymore. And so that takes you to the barium plus plus state in either liquid or gas barium tagging experiments. Now in gas, that's the end of the story, but in liquid phase or in any dent in the solid phase, presumably as well, electrons which have thermalized around the track now recombine with the ions, and so you end up with a distribution of charge states between barium, barium plus, and barium 2 plus, whereas in gas you're left with a complete sample of barium 2 plus. So I'm not really talking about this today, but I couldn't not mention it, that our friends in the Nexo collaboration work on barium tagging in the liquid phase, and here the target is those barium atoms and singly charged barium ions. Now for those, you have outer electrons in these outer orbitals that you can cycle around with laser fluorescence. And so what they do is they use lasers tuned to the atomic resonance frequencies to cycle those around and detect the ions, usually in a thin layer of xenon ice. It's very interesting. The electronic environment in the solid xenon ice gives you these signatures that depend on the site, and they've shown some sensitivity to both ions and atoms now at this point, singly charged ions and atoms. It's a vibrant ongoing R&D activity at the interface of atomic and nuclear physics, but it's not at the interface of chemistry, which is what I was asked to talk about today. So the question then for us in the gas phase is, how do you make the barium dication shine when it has no outer electrons to cycle through atomic fluorescence transitions? And so the idea here is to add those transitions by hand. You can use this technique, single molecule fluorescent imaging, which involves taking a molecule which is designed such that it's not fluorescent when it's unbound to an ion, and then it becomes fluorescent when it becomes bound. And so the way this works actually is you have a dye, which is this piece here, and this particular molecule, this is called fluorescein. And this dye, if it was on its own, not bound to this molecule, you would excite it with laser light, and then it would de-excite and shine back to you like a normal fluorescence process. But this nitrogen has a special role because it has a lone pair of electrons. So when it's in this molecule, if you excite this dye, the electrons from this nitrogen dive into the now unoccupied ground state orbitals here, and they prevent the fluorescence transition. They switch it off, they quench it, if you like. So this story changes when you add an ion here, because this ion now goes into a bonding configuration in this cage, and the charge of the ion pulls the electrons from the nitrogen, which acts as a switch, into kind of a bonding configuration. So now when you excite this dye again, it will de-excite as you would expect it to, without being quenched by electrons moving within the molecule. And this is basically how all of these single molecule fluorescent imaging sensors work. They were mostly explored in chemistry for calcium sensing, because that's of biological interest, but calcium and barium are chemically very similar. And so the molecules which were developed for calcium sensing also respond to barium, which was one of the things we showed back in 2016 in this GINS paper. So the interesting thing about SNFI is it's a technique which has demonstrated single ion resolution. This figure on the left is showing you calcium sensing with single ion sensitivity in rat brain cells. And the bottom figure is showing you individual proteins and individual protein receptor sites being tracked with each one of these dots, green or red, as a single molecule. And they're tracking the chemistry inside a tumor cell at the single molecule level as these things bind or unbind to the respective sites. So it's a powerful method with single molecule sensitivity, and so it seems appropriate for single barium ion tagging. And these methods, I should add, won the Nobel Prize in Chemistry in 2014. So our work then, we wanted to adapt this for single barium ion sensing. And the first version of the setup that did this was a total internal reflection fluorescence microscope. The idea here is that you reflect light totally internally off a surface, and that excites a thin layer above the surface where your sensors and ions are situated. And with this kind of technique, we can take images like this one, which I imagine it won't play as well over the Zoom as it would if I was there in person. But hopefully you can see a sort of orange field with bright spots that are flickering on and off. These bright spots, each one is an individual barium ion. This is in a sample that we've prepared with these fluorescent sensors and then added a very dilute barium salt to it. So it's added a bunch of ions to the surface, and you're seeing the individual ions detected here up against the turf scanning surface. I'll upload the slides later if this video is not playing so well remotely, but hopefully you can at least see the spots. Now, the spots, if you zoom in on one of them and look at pixel by pixel, you get images like on the left. And the way that we know that these are indeed single ions that we've detected is that the flickering, the on-off behavior, if you have a single ion, when that fluorescent site decays through some chemical reaction, usually with local oxygen or something, it decays in one step. If it was like a cluster of fluorescent activity, it would die and bleach slowly and surely. But these single ions, they have single chemical reactions with a single molecule, and then this single step photo bleach is kind of the hallmark of a single ion detection. And so this was one of our hallmark papers. We detected single barium ions with 2 nanometers spatial resolution, which is significantly smaller than the wavelength of the light used to probe them with, and about 13 sigma statistical significance. Okay, so now onto the real chemistry stuff here. How much time do I have left, by the way? Can you give me a time check real quick?
Right, this was 15 minutes, so halfway.
Great. Okay, so the program that we pursued now was to adapt these kinds of molecules so that they work in the environment in which we're really interested. So this is the molecule I introduced to you before. This is actually a very famous calcium sensing molecule. It's called fluo3, and there's a variant of it called fluo4, which is just like this one, but with fluorine instead of the chlorine, and it makes it slightly more photostable. But this actually fails to work in our environment of interest for 2 key reasons. The first one is that this fluorescein fluorescent group doesn't shine in the dry phase. In order to undergo fluorescence, this group, this chemical has to change shape. And that only works in the solution phase. When it's inside like a solid matrix or tied to a surface, it can't really rearrange its configuration in order to get in and out of the fluorescent excited state. So it doesn't shine in the dry. The second thing is that the binding group here that cages the ion, you see it has these 4 carboxylic acid groups. This CO2H is a carboxylic acid. So the reason this is an acid is because the H can leave and be H+ when it's in solution. And so you see over here that those H's have left. This is the COO. The H's have left. And so this is like a negatively charged claw that can grab the ion. Now that also is not something that happens outside of aqueous solution. Acids are only acids when they're in solution. They're not acids in the dry because the hydrogen can't leave when it's not solvated. And so this molecule, which is demonstrated in the previous slides, is both made of a fluorescent group that won't work in the dry and a binding group that won't work outside of aqueous solution. And so we had to build our own molecules for the purpose of dry barium ion sensing in double beta decay. So this is at the point where we initiated a collaboration with organic chemists at UTA. And so we published in Nature Scientific Reports recently the first family of molecules which work in the dry phase for barium sensing. The old molecule is this one on the left, the fluoro3. The new one actually looks much simpler, in fact. It's based on now a crown ether group. So this just has oxygens that have lone pairs of electrons, so they can grab the ions in a neutral kind of bond instead of having to be charged like this. It means it's a slightly less effective grabber, but it's effective enough for our purposes. And then this fluorescent group here is anthracene, which is a rigid fluorophore that doesn't change its conformation when you excite it and de-excite it. And so then this nitrogen acts as a switch, the same way that this nitrogen over here did. So this was the first class of molecules that were then able to show this dry phase fluorescent response to barium. Actually, I'm skipping over lots of steps in the story here. It's very interesting, but I only have a half an hour talk. But these molecules turned out to be non-ideal because anthracene requires ultraviolet excitation. And when you're working at the single molecule level, everything is fluorescent. The glass, the microscope optics, everything is a bit fluorescent. And things are much more fluorescent in the UV than they are under visible excitation. So we quickly, after publishing this class of molecules, advanced to this class, which are now based on a visible excitation and visible emission fluorophore. This is naphthalamide now, this fluorescent group down here. So this one on the right-hand side is kind of our state of the art. It's dry sensitive, visible excitation and emission, barium selective fluorophore, 18 crown 6 naphthalamide. And there's also a publication, which I'll mention in a minute, that goes with that. So to really tune these molecules up and make them do what we want, you can either just experiment with 100 different kinds of molecules on a wet bench, or you can do precision molecular design. And we've kind of mixed both approaches, I would say. We've reached the point now where with density functional theory calculations, we can predict which molecules will respond to which ions and how strongly they'll respond, and also explore the different fluorescent behaviors in the different cases. And it's all a question of how the energies of the fluorescent orbitals and the quenching orbitals rearrange themselves. And so this is an example from the most recent paper that's showing how we predict no fluorescence in the case of the unbound molecule, because there's this lone pair group that can de-excite into the HOMO after it's been excited. When it becomes bound to barium, the orbitals rearrange themselves. This one drops in energy, and so now the fluorescence transition is allowed again. And indeed, we predict what the data shows: no fluorescence with potassium, and then fluorescence with mercury. So this gives us a nice tool to really explore more broadly this space and optimize these molecules with more precision. Tuning selectivity is actually very important. We want this to respond to barium and certainly not to xenon ions, for example, because if it responds to xenon ions, it will be overwhelmed in the detector with background from ionized xenon. But we'd also like it not to respond to any other kinds of ions, because there's always a limitation of how cleanly you can prepare any surface. So there's ions everywhere, in the water, in the chemicals, in everything. And to get a really sensitive probe, which is not overwhelmed by background, you really want it to be as sensitive as you possibly can. So one of our major focuses in the last round of chemistry studies was to really make this specific to barium. And so it's this 1E is the one that's very barium specific. It shows lots of response in fluorescence to barium and then very little to everything else. So I've cut off the names of the metals along the bottom here. But this one on the right-hand side is barium, and then there's all the names of the other metals, monocations on the left and dications on the right. So this is now going to sound like really sort of technical mumbo jumbo, but it was a major technical advance. One of the things that people usually do in SMFI studies for single molecule imaging is the first thing they do is they wash the sample they're trying to image with something that absorbs all the free ions and doesn't fluoresce. And then you add the dye, and so you start to see this competitive reaction between the ions which are bound to the non-fluorescent thing and the ions bound to the fluorescent thing. And that's how you suppress the residual ions so they don't just overwhelm you with background. So it's actually pretty new to be able to do single molecule fluorescent imaging without any competitive binder. And it's what we need to do for barium tagging, because we need it to be that when the barium lands on the surface, it doesn't go to some other molecule, it goes to this molecule. So we need to remove the competitive binder, which typically is BAPTA, this thing I've shown here. And actually with these tuned selectivity molecules, we were able to do that. We were able to take the first barium ++ ion images with no competitive binding agent in the system. So this is shown bottom left is a dry single barium ion. Here's the typical photo bleach showing it's a single ion, and there's the field of view showing a few of them. And this is all the barium ions in the sample now. This is not the small subset which emerge over the background from the bound ones in the competing group. I wanted to mention that here because it's actually a major step, even though it's a little bit hard to explain. Okay, so obviously this is a wide field. Sorry.
You have 7 minutes left.
7 minutes, that's great, perfect. So obviously this is a wide arena to explore. There's many different kinds of molecules with many different kinds of behaviors that you can imagine. And it's kind of started a little bit of a cottage industry, actually. Now there is a group working in Spain developing new classes of molecules for barium tagging. Our collaborators on next. And this particular advance I'm showing here is unlike our molecules, which go from being switched off to being switched on when they grab barium. This one goes from being emitting in the green wavelength to emitting in the blue wavelength. And so it's like a color change rather than an on-off molecule. And this is like another promising approach that you could adapt to the barium tagging idea. But there's a lot of space to explore with these kinds of fluorescent sensing molecules. Okay, so now I would say that the next major challenge is to pack the surface density with these things, because those images I showed you before were all taken in samples with fairly sparse coverage of fluorophores. What we need to do eventually is have a surface where the barium is going to land in a capturing receptor if it hits the surface. And so what that implies is some degree of surface density and possibly also some degree of ion mobility across the surface when it gets there. Those are both things that you can tune in these chemistries. And the way we're approaching this is to try and grow a self-assembled monolayer, densely packed self-assembled monolayer on the surface of these molecules tethered down with an anchor. The real challenge, I think, is that when you put fluorescent groups close enough together, like you pack them this close together, you start to get collective effects. Their orbitals, they stick together like sandwiches or form hexamers, or their orbitals overlap with each other. And so preparing a dense monolayer without interference between the fluorescence groups is the next challenging frontier. I think the most likely way we think this is going to work is to have the molecules dotted around with spacers, but the spacers in between them have to be somehow ion conducting, so the ion can move along the surface. This is all stuff that can be done, but it's obviously a big tuning exercise to make it work well for what we want. But this is our present focus in the chemistry right now. I should also mention that we've been able to image single barium ions in high pressure gases. Obviously, this is the working environment that we care about, is imaging these things in high pressure xenon. This system at the left is a high pressure microscope developed at UTA. I think it's the first one of its kind anywhere. And the image at the right shows a barium dication imaged with single molecule fluorescence imaging in high pressure xenon gas, which is the first time this has been done in the working medium of a double beta decay experiment ever. So this is a very promising thing, but these pictures can be slightly misleading because they maybe sort of convince you that we've solved the problem already. But the key issue here is that this is a non-densely packed array, and we need to have a fully dense fluorophore array in order to capture the ions efficiently everywhere on the surface. Okay, I know that I was supposed to be talking only about how we've absorbed chemistry into high energy physics in this workshop, but I also wanted to talk about a couple of our spin-offs that we're quite happy with. So we're developing these novel chemistries, and actually we're studying fluorescence detection of ions in ways that people don't usually think about it somehow. One of the things that we've done in the process of this optimization and exploration of molecules is we accidentally invented what we call the mercury monster. This is a molecule which has world-leading sensitivity to mercury, and it's also unconfoundable. So if you add different metals and mercury, this thing will still respond to the mercury rather than responding to the other metals. So it's very selective and very responsive to mercury. And it also is non-fatal when it's in living cells. And so the pictures at the bottom here show you mercury at the, I think it's nanomolar concentration in some cancer cells. A is the top, and D is if there's no mercury, which have been injected with this molecule. And you see the sort of bright field in the overlay on the right. And we'll be publishing this soon, because this actually has very interesting applications for in vivo or ex vivo mercury assay at record low concentrations without cell mortality, which is very nice spin-off from this work. The other spin-off that we're now developing is more specifically about the gas phase sensing of ions. So it's been well known for some time that long-term exposure to atmospheric metals is something that correlates to mortality. So in short, if you live somewhere where there's lots of metals in the air, then you're likely to die younger. And this has been assessed in various places. This is a paper doing it in France. There's one in China, and I think there's one in Portland, Oregon as well. I don't know why those places specifically. But the way that this is usually assessed, the airborne toxic metal concentration, is kind of a crude way. Is that you take a bunch of moss off the local rocks and then dissolve it in some concentrated acid, and then mass spec the moss, because the moss has absorbed metals from the atmosphere. It's because this sort of gas phase airborne chemistry is not a very standard thing to do, really. So we're developing now real-time airborne toxic metal sensing via fluorescence at the gas-solid interface, essentially applying the techniques developed for barium tagging, so that we can sense in real time rather than waiting for the moss to accumulate the metals and then assaying it. And this is work that we've just recently got funded by the NSF and the Welch Foundation. I should also say that the barium tagging work is sponsored by the DOE. Everything I've showed so far is DOE supported, but this particular piece of work is NSF and Welch Foundation supported. Okay, so I think that's all I wanted to cover. So high pressure xenon gas is an especially compelling technique for neutrino-less double beta decay searches due to energy resolution, topology, and scalability. It also appears to enable possibly barium tagging. This would give us a major increase in sensitivity. Single ion imaging with single molecule fluorescence imaging shows great promise, and barium ions have now been individually imaged in high pressure xenon gases. A broad field of chemistry still exists to be explored here, including fundamental open questions in chemistry about chemical reactions at surfaces and in dry conditions. Thank you.
Thank you very much, Ben. That was super interesting. Are there any questions? Please raise your hand in the blue hand.
Can I say one thing while I'm waiting for people? We're looking for a postdoc to work on this. So if any of you are thinking about a postdoc or know one, then tell them to talk to me, just while people are thinking of their questions.
I don't see any hands. If you don't know how to raise your hand, just speak up, maybe. If I may, I had one question. Can you say a little bit about the time sequence of the fluorescing? Are these very short? What's the time scale of these activities?
Yeah, so let's see, do you mean this plot at the bottom middle here? So that
yeah.
This takes, so once it's grabbed an ion, in these conditions, it takes about 1 minute to photo bleach, but that actually depends on your laser power. So if you drive it with, it's like basically accepts a certain number of photon cycles before it dies on average, because the time when it photo bleaches is when it's in the fluorescent excited state, because that's a chemically reactive configuration of the molecule. So if you never excite it, it basically probably will live approximately forever. But if you excite it frequently, it will be in that chemically reactive state more often, and it'll die sooner. So I think this is 2 milliwatts of laser power causes it to die in about 1 minute. This is fine for this R&D. I think the laser power is something we can tune, of course. We can always get a more or less powerful laser. So it's sort of up to us to decide how long it lives.
Okay, thanks. So there's a question on the chat from Anil. How are the barium molecules stable in terms of time? I guess you just answered that. James Buckley?
Yeah, I just wanted to follow up on the last question about timescales. There's also the ability to use fluorescence lifetime as, a probe of chemical environment, but maybe also to discriminate the actual signal fluorescence from some background process. So if you have a very fast pulse laser and you measure lifetime on sub-nanosecond or nanosecond kind of timescales, you can often do chemical discrimination. So I was kind of, in addition to the photo bleaching time, there's this fluorescence lifetime information. I was wondering if you could combine that in any useful way.
Yeah, we're aware of it and we've thought about it. We don't have that laser. In fact, we have a laser which is not fast enough. Do you have that laser? Like we're very interested to work with people who have
no,
these tools.
I can tell you, I collaborate with someone named Matthew Liu, who came from Murner's group, the Nobel Prize winning group and at Washington University in St. Louis, and he basically does this kind of thing. There are other people, I think, at the University of Illinois and elsewhere that do fluorescence lifetime imaging. So I would contact those people.
Yeah, I think we should. I mean, there's so much to study here, and as you point out, there's a lot of things to study here that are already well known by people outside of our community, right? Where they can probably do those kind of studies fairly quickly and find out if it's interesting. So, yeah,
Right.
Thanks. I'll follow up with you offline.
Okay.
All right, so I don't see any other questions. Thanks again, Ben. This was very interesting. And we can move on to the next talk, which is by Francois Leonard from Sandia National Lab, and about novel room temperature photon detectors using carbon nanotubes. And so you have 20 minutes total, including question. I will signal you after 13 minutes.
Great. So can you hear me and can you see the slides?
Yes. Maybe, can you make it full
I
screen?
am trying right now. How's that?
It looked black to me, and now it's still showing the outline.
Let's see. Try that again.
I only see your camera.
Yes.
It's fine. Otherwise, we stay with the other view.
Trying it now. So
Yeah.
you're seeing,
Yes.
yeah. So I'm just going to leave it like that and not project a PowerPoint, which seems to change things. So, all right. So what I want to talk about this morning is harnessing the nanoscale for HEP scale research, ? So my name is Francois Leonard, and I'm from Sandia National Labs in Livermore, California. So the plan of my presentation today is to try to capture some of the spirit of the workshop and talk about collaborations. And one of the important aspects of collaboration is knowing who your friends are and what they do and what they bring to the collaboration. So I'll talk briefly about who we are and what we do. Then I'll give you an example of a recent collaborative project that we've done. And this will lead me into posing a question in terms of potential collaboration related to HEP. So about who we are. So I'm at Sandia National Labs.
Still see the title slide.
You still see this title slide.
Sometimes on slideshow.
Not sure I'll fix that.
Yeah, sometimes the full screen is weird, so maybe I would suggest just don't use full screen.
Yeah, maybe it's not worth.
I was not. I was not.
Oh, I see. Okay.
So are you still stuck on the slide, the title slide?
Yes.
Okay.
Maybe just go back to what you had in the beginning.
Yeah,
Yep.
another option is to just share the whole desktop.
Yep. So now are you seeing another slide?
Not yet.
All black right now.
Yes, now.
Okay. Yeah.
All right. Okay. So yes. So I was trying to tell you about who we are. So I'm from Sandia, and we have two main sites. One is in Albuquerque, New Mexico, with about 10,000 employees, and another one in Livermore, California, where we have about 1,200 employees. And that's where I'm located. And Sandia has many missions, but our work is in nanoelectronics and nanophotonics. And so what our group does is it looks to both discover and exploit the unique properties of nanomaterials for electronics, photonics, and optoelectronics. And so the one aspect is just to understand the basic science, fundamental properties such as electronic and photonic of the nanomaterials, but then use them for some functionality, such as in electronics or photonics. And so just to highlight the collaborative aspects, so we have several staff members, postdocs, and students that have very varied backgrounds, including physicists, material scientists, chemists, electrical engineers, chemical engineers, and so forth. We love to collaborate, so we have collaborations with universities, other national labs, as well as industry. And these collaborations are often enabled through the various sources of funding that we have. And so we have some internal programs at Sandia and within the NNSA for collaborations. We have funding from DARPA, where we always have to build sizable teams. Same for the DoD. And of course, as a DOE lab, we also obtain funding from the DOE Office of Science. And this is through, for example, the Office of Basic Energy Sciences, and particularly on the material science part. And then we also have projects with EERE. So just to give you an idea of the type of multidisciplinary research teams in the group. So first of all, one thing that we really strongly value is a close collaboration between theory and experiment. So this is something we always try to build in our projects. So we have efforts in nanoelectronics, where we both model, fabricate, and test devices with individual nanotubes, or such as a gallium nitride nanowire in this case. We also have extensive work around photodetectors. So this is an example of a solar cell we made using core shell nanowires. I'll tell you more about detectors with carbon nanotubes. And recently, we've been looking at topological materials as well. We also have an effort in printed electronics. This is an example of a terahertz sensor that we made by inkjet printing carbon nanotubes. And we were able to do terahertz imaging at room temperature, such as looking at the internal structure of this butterfly wing. And finally, with all the interest in artificial intelligence, we have a number of efforts right now looking at making individual devices for analog computing, as well as looking at optical neuromorphic computing, where we simply process light fields in order to extract features from images. Okay, so with that, what I want to do is I want to talk to you some more about a particular project that involved a larger collaboration that we recently executed. And this project was funded by DARPA and is about fundamental limits of single photon detection. And the motivation for this is that, of course, single photon detection is pervasive across science as well as many applications. And currently, there are different technologies that are able to detect single photons. So you look at superconducting nanowires, avalanche photodiodes, PMTs, and remarkably, even the human eye is able to detect individual photons. So the question is, for example, given that nature and humans have been able to construct these sensors, are there fundamental limits to single photon detection, and what determines those limits? Another question is, are there trade-offs between metrics? So if I have very high efficiency in the detector, does this mean that it inherently will be slow, or that the jitter will be high, for example, and what determines those metrics? And another question is, what is the best architecture for a photodetector? Is it something like the human eye, or do we really need something with a phase transition like a superconducting nanowire? And finally, a cross-cutting question here was, are fully quantum coherent detectors advantageous for single photon detection? And so in order to address this question, DARPA and then our team was composed of a broad range of folks with backgrounds in physics, material science, chemistry, and in this case, it was really critical to engage with quantum theorists. And so the approach was first to develop a new general theoretical framework for photodetectors, and then use some fabrication and testing of these to try and assess whether these new designs could be valuable. So the first aspect is that we were able to realize a new general modeling framework for photodetectors that is from the ground up and basically uses techniques from quantum information science and quantum optics, so that this general framework can handle a really broad range of photodetectors. And so the photodetector could be composed of atoms, molecules, it can be more extended solids. We can handle this very broadly. You could have a single detector or several detectors that are all interacting with the photon field. So we can handle some pretty general situations. And importantly, what this framework can do is it can generate performance metrics. So in other words, from the basic tools of QIS and quantum optics, we can calculate the efficiency, the jitter, the count rate, and so forth for these detectors. And it's also practical in the sense that if there's a type of detector or an architecture for a detector that you're interested in, we have a way in which we can map this into our framework and assess its potential for different types of applications. So I want to give you an example of using this as a way to design a completely new type of photodetector for a functionality that is extremely difficult to realize with existing detectors. And that's the ability to do photon number resolution. So suppose that you have a pulse of photons that contains, let's say, 1 or 5 or 12 photons. And what you would like to be able to do is to tell exactly how many photons are in that pulse. So that's very difficult to do with existing detectors. So the new construction that we came up with consists of a very dense array of individual nanoscale sensors, all within the wavelength of the photon. And you can do this either in 2D or in 3D. And the main thing is that all of these sensors are simultaneously, coherently interacting with the photon. So with this, you can realize in principle some remarkable things. So first of all, you need to design the architecture for each of these elements and how they interact with the other elements in the photon. And this is just a very high level representation of this in how we can structure the energy levels for the photon absorption and the transfer of energy through a system, and then monitor this, for example, with an amplifier. So of course, this is high level. It allows us to establish the basic physics, but of course we want a physical realization. So this is an example of how we took this type of high level architecture and mapped it into a physical system. And the system consists of carbon nanotubes that are functionalized with C60. And then there's another layer of a molecular system on top of this that serves to absorb the photon and transfer its energy to the C60. So the performance of this system is actually quite good. So for example, suppose that we require a 99% efficiency of detecting up to 12 photons. So what we've shown from our calculations is that we can achieve this very high efficiency while also having a count rate that is on the order of 0.5 gigahertz and a dark count rate that would be less than 1 per year, okay? And I put this line here because I think this is relevant to HEP. So for example, for dark matter searches where very few events are expected, there's existing work in using superconducting nanowires for that purpose. And of course, you want some very low dark count rate. Okay, so that was an example of using techniques from QIS and expertise from quantum theorists to design new detectors. And then the second aspect is to actually start to realize these experimentally. So we had an effort to fabricate these detectors. So this is an example AFM image of an array of aligned carbon nanotubes connecting 2 electrodes. These sit on silicon dioxide and there's a backgate, so this is really a transistor. Then what we do is we functionalize these nanotubes with different molecules. So as an example, we functionalized the nanotubes with azobenzenes. When a single photon comes in, it triggers a conformation change in the azobenzene, which changes the current flow in the nanotube. And with this, we're able to demonstrate at room temperature a gain of greater than 10 to the 4. Now, because of the noise in these systems, we wanted to improve this gain even further. So we went to a different molecular functionalization, in this case, C60. So the C60 is simply evaporated on top of the nanotubes. It's a very, very thin layer, about 10 nanometers. And in that case, we observe extremely high sensitivity. So this is an example of a photon pulse coming and triggering this detector. And the gain at room temperature for the system is greater than 10 to the 8. In case you're not familiar with gain, this means that every photon that comes in is affecting 10 to the 8 electrons in the carbon nanotube. So this is in array form. And what we did is we made devices with only a single carbon nanotube, in which case we were able to demonstrate the detection of 200 photons at room temperature. And there's a lot of room for improvement in those devices as well. And so finally, the last thing that we've done is replace the C60 molecule with P3HT. And this system is even more sensitive, greater than 10 to the 9 gain at room temperature. And we demonstrated the detection of between 8 and 13 photons per carbon nanotube, which is about a factor of 5 better than what we achieved with the C60. And so we anticipate that scaling this down to devices with fewer nanotubes would further improve our sensitivity. Okay, so what I've shown you so far is an example of combining expertise, from chemistry, device fabrication, quantum modeling, and so forth, to address this broad problem of single photon detection. Now, what I want to do is show you how we've been doing some work to take advantage of this to scope out a problem that we think is relevant to high energy physics. And that is
6 minutes left.
Yes. And the problem is to determine the color of a single photons. So you want to be able to do this in a detector that maintains very high efficiency, that has low jitter, and then at the same time, you want to be able to have very high energy or wavelength resolution. And so this is, again, something that doesn't currently exist. And the question would be, does fundamental physics allow you to do that? And if so, how would you design such a detector? And so why do we think this is relevant to HEP? So this is work done with Andrei Ilagin at the University of Chicago. And the idea is that in liquid scintillators, in order to reconstruct trajectories, it would be very useful to know the wavelength of the photon, since the speed of propagation at different wavelengths is different. But the requirements are very stringent. So for example, you need less than 10 picosecond timing resolution and less than 10 nanometer wavelength resolution. Okay, and as far as I know, there's no existing photodetector that can do that. So how do we proceed to kind of scope out this problem and see if this would even be feasible? So what we did, as I said before, is we used tools from QIS to come up with a new design that I'll tell you about in a minute. And we looked at some performance projections. So let me tell you about some of those results. So the design is very similar to what I showed you earlier, where we have multiple nanoscale elements all within the wavelength of the photon. The key difference here is that each element is sensitive to a narrow band of wavelengths. So when the single photon comes in, it's going to simultaneously interact with all of these elements. Some of these elements are more resonant with the wavelength of the photon, and it will tend to trigger those elements. Then you want to read out those elements using an amplifier. And so each of these amplifiers then corresponds to what we call an energy bin, where we're resolving the photon. So again, using the techniques that I introduced earlier, we've calculated the performance metrics. So here's a plot of the efficiency for single photon detection as a function of these different energy bins. So in this case, for example, we sent in 3 photons, a yellow photon, a green photon, and a bluish photon. And you see that the yellow photon gets detected with very high efficiency, in the high 90s, and only triggers bins that are in a very narrow distribution around the bin that we had previously said was the yellow bin. So similarly, the green photon can be detected very precisely and doesn't overlap with the yellow photon. And then the blue one, which we don't care about because it falls outside of our detection window, you can see doesn't pollute our energy bins. And so this shows in principle that physics will allow you to do this very high energy detection, energy resolution detection, while also maintaining high efficiency. And didn't have time to go into the details, but this requires some very careful engineering of the energy levels and the distribution and the number of these absorption systems. So now we have high efficiency and we have potentially high energy resolution, but for the problem that I was mentioning, we also need low jitter in order to get good timing resolution. So from our approach, we were also able to calculate the jitter in this system as a function of the energy resolution. And the first thing that you see is that there's a trade-off, right? The lower that we want the jitter, the price we have to pay is that the energy resolution will have to be higher. But the numbers are actually very good. So if you look here at my lowest energy resolution of about 40 MeV, we still only have a jitter of about 500 femtoseconds. And this energy resolution corresponds to about 10 nanometer resolution in the visible. Okay, so for us, this is exciting because it shows us that there's a path to an even other type of detector that achieve a functionality that is not currently achievable with existing detectors. Okay, so just in closing, I hope that what I was able to show you is that fundamental physics gives very few limits on photodetector performance, and that we think that there are new developments that are possible with nanoscale elements that enable new levels of performance. And I've shown you that we've already begun testing some of these designs and achieved some quite remarkable gain at room temperature. And finally, we've identified what we think are interesting benefits for HEP. And maybe my main message this morning is that, like I said, we love collaborations, and we are very much interested in working with folks in the HEP community to identify additional opportunities, help form teams, and develop proposals. So thank you for your attention.
Thank you very much, Francois. This was very interesting. Are there questions? No. Stunned silence. Is your group only working on photo detection, or also other things related to nanotubes?
Yeah, so like I was saying in earlier, we have a number of efforts in different area on nanoelectronics, for example,
Okay.
different types of devices like transistors, or it could be diodes, or and things related to artificial intelligence as well. So
That's
for
all the
analog
same,
devices.
all the same group?
Yeah.
Okay, thanks. Any other questions? Does not look like it.
So I would encourage people to submit questions. We do have a panel later to submit
Yes.
For the panel.
All right, then we can move on to the next talk. Thank you. The speaker is Alexander Millar from Stockholm University, talking about quantum materials for axion searches.
Hi, so just getting up my screen. Hopefully you can now see my screen.
Yes, let's see if it goes beyond the title slide.
Excellent. So thank you very much for virtually having me. So I'm just going to talk a little bit about the kind of new research environment that's focused on axions that we applied for a grant last year and received. So it's kind of covering quite a few complementary ways to search for axions using the kind of interdisciplinary nature of the expertise at Stockholm University. So just a small bit of background. The axion is one of the more well-motivated dark matter candidates, partly because you actually get to the axion by trying to solve a different problem from dark matter. So if you do a kind of a non-perturbative analysis of the QCD sector, what people found is that there is actually the potential for another term to exist, a CP violating term called a theta term, which is essentially an angle times the gluon field strength tensor. And this theta term, because it violates CP, it would actually cause a charge separation in particles like neutrons, and it would lead to an electric dipole moment. And while we have lots of neutrons around, so we've done very precise experiments, nobody's ever seen this charge separation. So it actually puts an extremely strong bound on this theta angle that suggests it has to be less than about 1 part in 10 to the 9. And a lot of people feel it's very unsatisfying to have a fundamental constant, which is, you know, arbitrarily close to a special value without any obvious explanation. So in the standard model, one interesting aspect is that the potential energy of QCD is actually minimized if you go to the CP preserving value. It looks kind of like a sine cosine potential. So one way of thinking about the axion solution, which is the kind of preferred solution to this problem, is to, the phrase is promoting theta to a field. So if theta was actually a dynamical field, then you would naturally go to the bottom of its potential, and then you would have a dynamical solution to the strong CP problem. So schematically, what that means is you introduce some new U1 chiral symmetry that breaks at some very high scale. And it doesn't really matter, after this is broken, you have some angular degree of freedom, which is left unbroken at high energy. And then when QCD effects become important around the QCD phase transition, the potential acquires this cosine shape, and the axion kind of naturally goes down to the bottom of its potential. But as there's some kind of interesting properties of pseudo Goldstone modes, which is what the axion is that its interactions are made conversely weak the higher up the scale of symmetry breaking. So if you have a very high scale of symmetry breaking, then the axion's interactions are very weak, and there's no real friction to these oscillations. But you imagine if you have a ball going down the bottom of its potential and there's no friction, it just keeps oscillating forever. And these oscillations, which are only damped by the expansion of the universe, actually behave exactly as cold dark matter. So it's kind of this nice 2-for-1 system where you've tried to solve the strong CP problem and you got a dark matter candidate as well. So that's kind of the background for this research environment. Historically, the axion has often been kind of considered the second most favorite dark matter candidate after WIMPs, but particularly in the last maybe 5 or so years, due to the very good WIMP experiments still not finding a definitive candidate, people have started to kind of broaden their horizons a little bit and seriously consider trying to have a comprehensive strategy to search for different dark matter candidates. And the axions has a kind of a phenomenological point that makes this a little bit easier, in that almost all of its parameters are determined just by the axion mass. So once you set the axion mass, its couplings and everything like that are all set up to order 1 numbers. And one of these couplings that's very interesting is actually that to electromagnetism. So the axion actually has some anomalous coupling to electromagnetism, and also via the mixing with the neutral mesons like the pion, which can be exploited because we have lots of different ways of exploiting electromagnetism in order to search for the axion. So recently, particularly in the last 5 or so years, there's been a huge increase in the amount of efforts in order to search the kind of entirety of this one parameter theory. So that's the kind of the background for this research environment, which started last year. So the kind of the permanent members are Stefano Benetti, Jan Conrad, Jon Gudmundsson, David Marsh, Haranya Perez, who is the PI, and she'll also be in the panel discussion later, and Frank Vilcek. There are also quite a few PhD students who have become associated with it. Alex Adler, Nadia Dakhlefri, sorry, I screwed that up. Katie Dunn, who you probably know from the organizing committee of SNOMAS, and Ike Mueller. There's also some postdocs like myself and Matthew and Kier, and now Tom Edwards, and quite a few people who are at Stockholm University who have now become kind of associated with the research center. So they're kind of additional expertise, even if their main area isn't on, say, axion physics. And this allows us to draw from much more kind of interdisciplinary pool than would be normally considered. So with all of that kind of manpower, the research environment focuses on 3 kind of key areas. So the first is looking for axions more directly. So like most dark matter candidates, you really want a smoking gun. So the equivalent of WIMP direct detection experiments in the axion world are usually called haloscopes, basically because they're kind of a telescope for the dark matter halo. And the first work package is exploring a newly proposed kind of haloscope called a plasma haloscope, and basically trying to work out the practicalities that would go towards realizing a full-scale experiment. Kind of complementary to that is indirect searches for axions. So the axion has kind of quite unique properties that leave very distinctive signatures in both cosmological and astrophysical environments. So if you can get some good hint of an axion from these sources, and I'll say hint normally because usually people trust indirect detection a little bit less than they trust direct detection. It would both be very interesting in and of itself, but it would give you somewhere to look with these experiments. So a lot of the features of these experiments is that it's much easier if you already know where to look than if you're kind of trying to search the entirety of this one-dimensional parameter space. The third work package, which maybe gives the talk its name a little bit, is using interesting condensed matter systems to search for axions. So basically using a novel interactions between axions and quasi-particles in order to potentially lead to axion detection. So there's kind of an interesting synergy between the three. So the work package 1 and 3 are kind of more focused on direct detection, and work package 2 is really focused on exploiting the sky for indirect hints. So the first one is kind of in some sense the most straightforward in terms of searching for axions. So some of you are probably quite familiar with axion physics already. So an experiment that's direct detection is usually trying to convert an axion to a photon in some strong magnetic field. And then you detect this photon, which you can see using, for example, a very low noise amplifier. And the kind of fundamental problem of axion detection is that the axion has a mass, and generally speaking, the photon does not have a mass. So you have to have some way of meeting energy and momentum conservation in order to allow this conversion to proceed. So the traditional way was proposed actually by Pierre Sikivi. So this is sometimes called a cavity heloscope or a Sikivi heloscope. And what that's essentially doing is breaking translation invariance. So once you have these cavity walls, the photon wave function isn't a plane wave, it's whatever the cavity mode happens to be. So you don't actually need to provide, you don't actually need to match the momentum of the photon to the momentum of the axion. Because essentially the cavity walls are providing the mismatch. So if you have some cavity which has a resonance frequency that matches the axion's frequency, then you can resonantly populate photons into the cavity mode and then detect them using some fancy low noise amplifiers, which actually themselves can be interesting quantum systems. So the plasma heloscope is in some sense a slightly simpler idea. It's to just give the photon a mass. So if the photon has a mass that matches the axion's mass, then you don't need to provide any extra momentum. They'll naturally have the same energy and momentum relation. And this idea of giving the photon a mass is exactly that of a plasma. So a plasma at the plasma frequency essentially leads to the photon having a mass equal to the plasma frequency. And if you can have some plasma which you can control its properties, you can tune that plasma frequency to match the axion frequency for a wide range and then scan through different axion masses. So this was proposed last year by Matthew Lawson, myself, a couple of people who are not directly in the environment, and Frank Wilczek. So one of the main materials we're looking at for this is what's called a metamaterial. So it's kind of non-trivial to find plasmas that have the right properties. So if you want to change the plasma frequency and operate at cryogenic temperatures, there aren't very many options. So for example, a hot gas, you can very easily control the plasma frequency by changing its density. But you can't put it at cryogenic temperatures without it turning into a solid. If you have a solid chunk of metal, that can be operated at arbitrarily low frequencies and still be a plasma, but you can't tune the frequency of a metal. So a metamaterial is a kind of a neat solution to this. So metamaterials are more generically just materials where the main material properties are determined by geometric concerns and not just the composition of the constituent elements. So a thin wire metamaterial is kind of one of the simplest metamaterials. It's just a series of very, very thin wires, which are spaced smaller than a Compton wavelength apart from each other. So photons can't resolve individual wires. They just have this kind of collective effect. And this leads to some kind of collective behavior just due to the mutual inductance of electrons in the different wires. So this is both obviously cryogenic, or at least friendly to cryogenic environments, because if you cool down wires, they don't appreciably change in properties. But also because it's a metamaterial, the effective photon mass, it only depends on the spacing of the wires. So you can, if you can change these wire spacings, then you can control what your plasma frequency is, and then allow that to search for axions. We're also considering a couple of other examples, but this is the kind of the simplest and maybe most elegant solution. So this is just an example of a kind of a projection for what a plasma heloscope would be capable of. So just to kind of give some background for how to read these kinds of plots, the y-axis is the axion-photon coupling. So the higher this number, the more strongly it couples to photons, and the x-axis is just the axion mass. So for the QCD axion, it's mostly a one-dimensional theory. So once you determine the axion mass, these two black lines throughout the middle of the plot, those are the kind of the benchmarks for the most typical axion models. Ca gamma is just some order one number which determines this coupling. So you kind of have a very well-defined target goal for your search. You want to be able to cover, you know, this model band in as wide a range as possible. So the gray bars are pre-existing experiments, so those are actual results, actual limits. And you can see that as of now, we've only searched a very small fraction of the axion parameter space. I mean, essentially a kind of a Bayesian insignificant amount of the parameter space has been searched. But the different colored boxes are the projections of various proposed experiments or future experiments that are aiming to expand this to cover a much wider range. So you should also take them with a little bit of a grain of salt because they're all projections and not actual limits. And you can see in the dark green, plasma heloscopes are potentially capable of searching a very large fraction of this high mass parameter space. So since then, there's been interesting developments. So far, we have been mostly treating these metamaterials as just an effective medium. But if you really want to understand the ways that an experiment might depend on real physical parameters, so for example, if you've tilted some of these plates relative to each other, some of these wires relative to each other, or you've misplaced a wire or something like this, it's very important to be able to have real simulations, including the actual wire nature. So this is an ongoing effort to really simulate these systems. Another very interesting development was that at Berkeley, we actually got the group of Carl van Bieber, who is a very well-known axion experimentalist interested in this idea. So they've been pursuing building a prototype device. This was a little bit interrupted by COVID because they started at the end of last year and then had to kind of cease all operations for a while. But what they've done is it's kind of hard to see because these wires are so small, but they've strung these kind of metal frames with many of these thin wires and then stacked multiple frames so that they can build up a material. And then what they're doing is essentially characterizing these materials, which has been going quite promisingly. And once they finish characterizing them, they can start playing around with engineering solutions to, say, adjusting the spacings of these wires. So for those who are interested, there was also a SNOMAS letter of interest written, which can be found on this link. So Jon Gudmundsson is the person running the simulations at the moment. So the kind of complementary work package to that is axion astronomy. So so far I've mostly been talking about QCD axions, so axions that solve the strong CP problem. But a lot of people are interested in a more general class of axions called axion-like particles. And that's where you don't necessarily solve the strong CP problem, but you still have some pseudo-scalar particle that exhibits a shift symmetry. And these are very popular in things like fuzzy dark matter or in string theory. One interesting property is if you get the mass of the axion light enough, is that its de Broglie wavelength becomes astronomically relevant. So, you know, almost to the scale of a dwarf galaxy. And because it's hard to have collapses when you're smaller than a wavelength, you actually suppress the formation of small-scale structure in the universe. So what Kia Rogers and Garcia-Sciveres have done in the last year or so is to, you know, take a very good look at the astrophysical data, particularly that of the Lyman alpha forest. So this is the kind of the imprint of the partially reionized universe, where you have the 21 centimeter line basically causing absorption that maps out the structure that the light is passing through as it gets redshifted. So by looking at this Lyman alpha forest, they're able to try and work out what the smallest scale structure is, and then that provides a bound on the axion. If, you know, the smaller scale structure you find, the heavier the axion has to be to provide a smaller de Broglie wavelength. And this bound is actually the equal strongest bound that's been applied to fuzzy dark matter in this way. So that's very exciting work that's already been done. Another avenue is to kind of cheat a bit. So before I talked about using a laboratory with a strong magnetic field to search for axions, but of course laboratories and strong magnetic fields cost money. It turns out that astrophysical objects can have much, much stronger magnetic fields than we can possibly achieve in a lab. You know, for example, 10 to the 8 Tesla magnetic fields in neutron stars. And just like in a plasma helescope, neutron stars generically have some plasma surrounding their surface as kind of an atmosphere. So you can have incoming axions falling into a neutron star, getting converted at the plasma frequency, and then exiting as radio waves. And it will be a very distinctive line-like signal that will be very hard to mistake for an astrophysical background. So with David Marsh and Tom Edwards and a few other people like Axel Gruber and Matthew Wilson, we're looking at kind of exploring what happens if you get dense chunks of axion dark matter falling into a neutron star, because that would give us a kind of very specific transient signal that you could then look for using existing data sources. So for example, at SETI, the search for extraterrestrials, they really care about looking for very high frequency resolution, interesting time signature signals. And while we're not looking directly for aliens, you could always reinterpret these surveys as a search for axions. So that's something which we're looking at doing quite soon. So lastly, though not leastly, because obviously it kind of influenced the name of this talk, there's the interesting quantum systems aspect. So many particle systems often exhibit collective behavior that looks like a particle, which is usually referred to as a quasi-particle. In fact, the plasmas I was talking about before exhibit this behavior exactly. A collective oscillation of electrons is called a plasmon in that context. And you can think about it, those systems, as the axion converting into plasmons. The work package 3 tries to take a more general approach to this. So what the aim is to work out which different quasi-particle excitations would couple to the axion. And once we have a good candidate for what kind of excitations we should be looking for, to search through material catalogs to identify interesting candidates. And once we have those candidates, to synthesize them and use the very nice terahertz spectroscopy lab of Stefano Bonetti to kind of characterize these materials and see if they're suitable for axion detection. And if they are, you can imagine synthesizing these materials and building some kind of interesting terahertz detector and essentially looking for axions being converted into these more exotic quasi-particles. So there is some, there aren't any papers out on this yet, but there are some kind of interesting preliminary results looking at multiferroics. So this was kind of maybe a very brief summary, but it was just to kind of give an overview of the different ways at Stockholm University we're trying to use the kind of disparate expertise to search for axions over a huge range of frequencies. So we're talking going up from terahertz, which is milli eV, down to 10 to the minus 22 eV. So really trying to explore this one-dimensional parameter space in as much detail as possible. And if you want to know more information, aside from these slides, there's also a website that you can find using this link. So it might be broken at the moment. Need to double check that. Thank you.
Thank you, Alex. Other questions? Yep, James.
Yeah, I have just a question and a comment. The first comment is I guess I have to do this, but there's an archive paper that my group has submitted on dense axion stars falling through the resonant conversion region in a pulsar magnetosphere, which I think is similar to the idea you're talking about. We were actually trying to see if we could explain some fraction of the FRB's fast radio bursts with this. So I just wanted to point that out. It's on the archives.
This is certainly similar. We were taking a slightly different approach, looking at, I mean, the literature is a little bit varied on these terms, but in the language of dense and dilute stars, we were looking at mini clusters and dilute stars rather than oscillatons.
Yeah, we were really looking at, at condensate kind of stars or something like that. But anyways, I was going to point out though that the plasma frequency condition is something one sees, in astrophysics and, a lot of different settings. So I like that you mentioned that. I think indirect detection always plays a nice complementary role. And then in terms of the plasma haloscope, I mean, the big problem we have, in experiments like ADMX is tuning and scan rate, and as you see, you can project sensitivity, but it just takes a lot of work to scan through it, at the relevant sensitivity level. So I'm not sure I totally see, how the tuning and scan rate is going to end up being for, tuning the plasma frequency rather than just the
tuning of the plasma frequency is not as big a challenge as you might assume, because what did we do? So in ADMX or something like this, you tune by essentially moving around these dielectric rods. In the case of thin wire metamaterials, you only need to change one direction in order to change the plasma frequency. It turns out they're still an isotropic material, even if the wires are not isotropically spaced.
Right. It's a photonic crystal with one lattice spacing is probably enough to tune the plasma frequency.
Yeah. And this is something which we've tested both experimentally, and it's also something which was theoretically predicted very soon after these were first, explored as a metamaterial. So the tuning is I agree the simplest tuning mechanisms are traditional cavity haloscopes, but there's nothing in this high frequency range which has an entirely trivial tuning mechanism.
Right.
Just because it's so much harder to go up to to those frequencies.
And the really neat thing is you don't have to combine signals from lots of small cavities, and still maintain your form factor. So it's a really cool approach in that respect.
So, I'm not going to guarantee it's the best approach at those frequencies, just because, my PhD was on MadMax, but I think it's such an interesting area that you really want as many options as possible. Because at the end of the day, it's very correct that it comes down to practicalities. So if we can provide different ideas which have different practical implications, something that's a deal breaker for one method may not be a deal breaker for another. So hopefully at least one of those methods gets there.
Thanks.
Are there any other questions? I had one maybe on your last slide, if you can, on the third topic.
Oh, sorry, this slide.
Right. So for the new materials, not sure I understood completely. Do you first come at it from a theoretical side? Or you
So we're starting off with a theoretical side, just from the point of view that you need to have some assurance that it will actually couple to an axiom.
Mm-hmm.
You know, that's the, you know, it doesn't make a lot of sense to spend a lot of time synthesizing and characterizing material and then turn around and said, well, actually, if you had bothered doing the calculation, it does nothing.
Sure.
So,
Yeah.
you know, the first stage is really identifying what categories of materials are actually interesting. And then you can start narrowing things down a bit.
Thanks. Any other questions? If not, maybe we can move on to the next talk. Thank you. The next speaker is Patrick Harrington from MIT Lincoln Lab, talking about superconducting sensors for coherent neutrino scattering.
Hello, all. Put
Hi.
my slides up on the screen. Please let me know if the slide doesn't change when I think it's changing.
Right. Yeah,
There's
it's
the
changing. Yes.
Okay, good. So I will be talking about superconducting sensors for coherent neutrino scattering. And also in this talk, near the end, I'll touch on how ionizing radiation can affect superconducting qubits. This is an experiment that was published in Nature somewhat recently from a collaboration between Will Oliver's lab at MIT campus and Lincoln Laboratory with Joe Formaggio's lab in the Laboratory of Nuclear Science. And to introduce myself, I received my PhD from Washington University just this past May. And my background is with measurement and dissipation with superconducting qubits. I've also been involved in the ADMX experiment collaboration, and I've worked closely with Jim Buckley, who's in the meeting today. So I started at MIT in June, and my role as a postdoc is to collaborate between Will's lab and Joe's lab, and to actually facilitate that collaboration with Lincoln Laboratory and fabrication possibilities there. And so the experiment and the ricochet collaboration that I'll talk about is an example of these collaborations. So for the next slide, my outline is that I'll talk about the ricochet collaboration in these superconducting sensors, kilometers for detecting neutrinos. And then after that, I'll discuss how radiation can affect qubit lifetimes. So the motivation of detecting neutrinos, the coherent scattering of neutrinos, is that neutrinos are hard to detect. They have a very small cross-section, and often with that, they impart very little momentum on the particles that we are sensitive to, like the nucleus of an atom. For example, the Super Kamioka detector has 13,000 PM tubes. It's a kilometer underground and has 50,000 tons of water. Now, that's not something that you could put in a truck and use to detect nuclear waste. But with a desire to make smaller detectors and more sensitive detectors, we can use a neutrino detection scheme that is has a much larger cross-section. And that is using coherent scattering of neutrinos, which has been proposed for applications of detecting heavy water reactors as well as nuclear waste. And so with this regime of detecting neutrinos, in principle, we could have something that fits in a truck. So what is coherent elastic neutrino scattering? This was a mechanism proposed by Friedman in 1973. And basically, the Broglie wavelength of the neutrino becomes on scale of the nucleus of an atom. And so there's a coherent interaction that scales with the number of nucleons in the nucleus. And with that, the cross-section of interaction increases greatly as the neutrino energy decreases. So there's actually a surprising turn where even though the neutrino energy is decreasing, we are getting much larger cross-section. And that cross-section is described in this equation here, where G is a very small quantity coupling strength, but the advantage here is that the strength of this cross-section scales as number of nucleons squared. And so what this translates to is that when neutrinos are less than about 100 MeV, we can get nuclear recoils that are on the scale of kilo electron volts. And this has been detected recently. So from its proposal in 1973, and it was detected in 2014 with the coherent collaboration. And at Oak Ridge National Lab, they had a spallation neutron source by shooting protons at a mercury target. And with a sodium iodide detector, they were able to detect the presence of muon neutrinos that came from a pion decay. And the great thing about this detection scheme is with a beam, they can get a background for moments when the beam is off and then see the signal when the beam is on. So that differential detection is being key for having the rare events of neutrino scattering. In the ricochet collaboration, there are a number of institutes and universities getting together who want to detect neutrinos from a nuclear reactor. Now, the real challenge here is that the energy is much lower than, say, from a spallation source, but the flux is much higher. So we're pushing into further deeper into the low energy neutrino, much lower energy neutrinos than were detected by coherent. And the proposed method is to use low temperature bolometers to do this. Here on the left, I'm showing a absorber I'll discuss in more detail. And there's a small chip that's leading off of it with a gold wire bond, and that's a transition edge sensor. So this is the bolometer we propose to use. And with the thermal link between the zinc absorber that heats up ever so slightly from a neutrino interaction, that increase in temperature can be detected electrically. And the proposed site for a nuclear reactor that we detect neutrinos from.
Sorry, we don't see the image that you're talking about.
Oh, so now the slides stopped changing. I have a backup plan to use a PDF. Are you able to see my screen? Oh, no, I stopped sharing. That's why you can't now. Are you able to see the?
It's black. It says you started screen sharing, but I don't see anything.
I apologize for these technical issues. An alternative way we could do this is I have these slides on the Dropbox for my group, and I know Joe is in the meeting. So if you give me a moment with these technical difficulties, perhaps I can get Joe to put these slides up.
Also go back to the PowerPoint, but not the slideshow, maybe? No.
No.
Nothing's coming.
I'll see if I can try to pull them up, Patrick.
Okay, thanks, Joe. Well, as Joe is pulling those up, I'll continue to describe what the ricochet collaboration is. We're proposing to use a nuclear reactor to detect neutrinos. And the site for the nuclear reactor is in Grenoble, France. It's the ILL iFlux research reactor. And so the plan would be to have a cryostat with a low temperature bolometer that is some short distance away from this reactor, where we could then detect few events of neutrinos from coherent elastic scattering.
I have the slides up now. I can attempt to share them.
You have better luck than I did.
All right, let's see.
Great.
And people
I'm
see
ready
that.
slide.
Okay, and Patrick, just tell me to advance if you need to go further.
Okay. Let's go to the next slide, please. And Joe, you can advance to fill out the slide. Thank you. So with the plot I'm showing here on the right is a calculated background for not the Grenoble reactor, but a different reactor, of how many events we would expect to see given the recoil energy from a neutrino hitting the absorber. And so where we are targeting is to have a few events, characteristic events for the detector setup, meaning that background detection is quite important. But luckily, like with the coherent experiment, we could have an on-off differential, a lock-in type measurement to characterize the background. And so that puts us at having on order of 10 events a day, and the recoil energy being at about 100 EV. With 100 EV, a simulated temperature fluctuation of our absorbers on order of 0.1 microkelvin. But what this means for current fluctuation in our detector is about 2 nanoamps. So with state-of-the-art technology, we would be pushing just a bit farther on the best performance transition edge sensors in order to detect such a signal. Joe, could you please change the slide? What this looks like schematically is that we have the absorber coupled to a transition edge sensor, and then we would use the fabrication abilities and microwave readout abilities that have been proven with superconducting circuits to do this detection. And so at Lincoln Laboratory, we can fabricate high quality factor aluminum resonators integrated with Josephson junctions. And we can also design and fabricate near quantum limited amplifiers, such as the traveling wave parametric amplifier. So with these tools, we hope to demonstrate the lowest possible noise floor in order to detect these very minute current fluctuations corresponding to neutrinos. So how a transition edge sensor works is that we need to translate a temperature fluctuation into an electrical fluctuation. Well, the way to do that is with a material that has a very sensitive resistance, a resistance that's very sensitive to temperature, such as a superconductor that is alloyed such that it has a superconducting transition slope that is ideal for our detection dynamic range and sensitivity. And so with a TES that's iridium platinum, as in this case, we can tune where that transition is. And then with a current bias, we can look at the change in current due to resistance fluctuations and have that modulate a squid. So Joe, if you could click forward just a few. Right at the end of this slide, we see that there's a schematic that is showing how a TES as a variable resistor would then change the amount of current going through an inductor. That inductor then changes the flux in a squid, which has a very sensitive IV curve. And so the thermal fluctuations of the TES then turn into a current fluctuation, and a pulse of the current would resemble a pulse of the temperature changes in our absorber. Please go to the next slide. And what we propose to do is quite novel for the absorber, where instead of having a dielectric like material or insulating material like germanium, we're proposing to use zinc as the absorber because it has high enough Z, large enough nucleus for neutrino scattering. But also zinc becomes superconducting so that the excitations of this material are not simply nuclear recoils and phonons, but nuclear recoils, phonons, and quasi-particles, which have a very long lifetime. And the effects of having high energy particles incident on the superconductor means that quasi-particles will be created and last a very long time. So hopefully that means we can use that for pulse discrimination and be able to rule out what are events from the neutrinos and what are events from high energy particles such as neutrons in our background.
Patrick, you have 6 minutes left.
Okay, thank you. Joe, could you go to the next slide, please? So a detection scheme using transition edge sensors can be done at DC, where we would look at the DC change in a squid due to these current fluctuations from the TES. But another way of doing this is at RF frequencies, where we are turning a DC fluctuation into an RF frequency fluctuation by coupling a squid to a resonator. And at the bottom right, I'm showing an inset view of one of these devices, where right at the end of the resonator is a squid loop, and that squid loop is coupled to a loop that is the inductor for the TES. And so if the current through the coil changes, it then actually shifts the resonator frequency, which we can detect using a microwave tone and see a very slight phase shift of that signal. Joe, could you go to the next slide, please? And that change in the resonator frequency is something we can detect by continuously probing the device and at its resonance frequency or near it. And when the current changes in the squid loop, it will shift the resonance. For the plot in the top, I'm showing the real and imaginary part of a demodulated RF signal. And a change in the resonance then translates into a very small phase shift of the probe tone. The challenge here is that we want to detect these phase shifts on a time scale of a temperature pulse or a current pulse, which is on order of microseconds to milliseconds. So having a low noise floor means we can discriminate the phase shift difference between one resonance at a certain frequency and another just by integrating a signal from microseconds to milliseconds. At the bottom left, I'm showing some preliminary results where we've created these resonators and show we can in fact shift the resonance based on how much flux is threading the squid loop. Go to the next slide, please, Joe. And a very important aspect of this detection scheme is to have a lowest noise amplification that we can of the probe tone before moving up to room temperature amplification, because the noise in the probe tone signal is due to quantum fluctuations near quantum fluctuations at this limit, and also from the temperature of our higher amplification stages. So in summary, using a traveling wave parametric amplifier would give us a great advantage to push down the noise floor to make sure that the noise temperature of our amplifiers is not limiting our readout rate. And this is the traveling wave parametric amplifiers is something that we have great expertise at at Lincoln, and we can develop high dynamic range amplifiers for this very purpose. And Joe, could you please go to the next slide? In the few minutes I have left, I want to make a nod to an experiment that's come out of the EQUIS in the MIT Lincoln Laboratory group and Joe's group, where they were actually able to demonstrate how environmental radiation from the laboratory does put a limit on qubit coherence times. And so these were some traditional qubits, transmon qubits, that were measured repeatedly and energized up to an excited state and then wait some time to see if it had decayed. And the effects of radiation were detected in this decay by seeing how radiation would create quasi-particles that cause these decay processes. So please go to the next slide. And the decay rate gamma of these qubit transitions depend on the quasi-particle population in the superconductor, as well as other effects. But the physics behind this is that the number of quasi-particles in a superconductor will depend on the square root of the power incident on that superconductor, the radiated power, which comes from cosmic rays in the laboratory, as well as radioactive environment from the concrete in the walls in the building. Please go to the next slide. And so the first experiment that was done was actually put copper 64 in the device packaging with the qubits and look at how the qubit lifetimes changed over the course of many half-lives of the copper's decay. And it was actually seen that at short times, the decay rate was quite low. And then after many half-lives, that decay time ended up settling out to be a decay rate of these qubits, depending on other factors. From these data, the constant here of how the decay rate depends on the radiated power could be found nearly empirically. So what was done to do this is to characterize what the amount of radiation was from the copper source using Geant4, and also characterizing other incident power from other effects in the environment. And so the fact that it was seen that radiation could directly affect the lifetime is I guess, not just it's not surprising result, but it had not been shown before. And so it is a great result to just see that the decay time does track with the half-life. And Joe, could you go to the next slide?
Sorry,
The other,
we're out of time. Can you?
okay, I'll wrap up quite quickly. The other experiment that was performed was to put lead shielding around the devices, and that blocks the radiation. So from moving this up and down 150 times, it showed that, in fact, the decay rate just from the lab's radiation is decreased by a very small amount. It's not yet limiting the growth of quantum technology, but it is something that would have to be considered if qubit lifetimes end up being hundreds of milliseconds. And that concludes my talk. So I apologize for the technical difficulties, but thank you for hearing about these two experiments.
Thank you very much, Patrick. Are there any, is there any quick question? Otherwise, maybe we can also keep it for the panel discussion later. We can go now, thank you. We can go to our last talk by Jim Moran from PNNL on ultra-low background materials to measurements and the environment.
Sorry about this. I'm trying to share the screen here. Oops. Hopefully that comes through.
Yes, thanks.
Great. So I have to preface everything by saying I'm really an environmental scientist, right? I'm coming at this maybe from a different angle than what some of the previous speakers have been coming at this. And I also want to say that this story is really one that's still in progress, right? I think we've laid a lot of the foundation. We have some of the initial steps that we've gone through, but we're not to a natural conclusion or ending point yet. But what I want to do right now is take just a couple minutes right now to look at this first slide. And there won't be any slide changes for a minute or two here, so we won't have to worry about that. But I want to grossly consider at the high level a very serious problem that we have right now in environmental science. It's a persistent knowledge gap. I think all of us, all the citizens of the world, have become increasingly concerned about our atmosphere, right? The composition of the atmosphere and how that changes in that composition are going to affect society, our planet, in everything involved with the Earth through different climate forcing scenarios. Obviously, one of the biggest concerns we have is the amount of CO2 in the atmosphere. We can measure the amount of CO2 increase in the atmosphere. We know it's going up. We're setting records on nearly an annual basis. But when we try to set up models to predict what's going to happen to that CO2, predict what all the sources are and what all the sinks are for that CO2 so that we can project, instead of just measuring what CO2 has been in the past, how can we project what that's going to be in the future? We see a wide range of different scenarios, right? Different models have very different estimates of where we're going to be in the 20, 50, or 100-year time span. And really what's driving some of those differences are key uncertainties about carbon budgets around the world. And one of the biggest uncertainties, and one that I've been thinking a bit about, is really the carbon budget involved with soil. So you might not think about soil a lot, so I'll cut right to the chase. The amount of organic carbon bound up in soil, surface soils, just in the terrestrial portions of the Earth around the globe, the amount of organic carbon bound up there is approximately 2 to 3 times the total amount of CO2 in the atmosphere, right? It's a very dynamic pool. Every year, plants put a lot of carbon into the soil, and microbes respire a lot of that. And what plants put in, a lot, but not all, but a lot of that carbon just goes right back out to the atmosphere. It's a delicate balance, and you can easily understand that with such a huge a pool in that soil organic carbon, if you perturb that pool just a little bit, you could either create a net sink for atmospheric carbon or unleash a net flux for atmospheric carbon, right? But yet, when you really think about it, when you get down to the basics, soil scientists, you can go to soil conferences to this very day, right? And soil scientists will argue and argue about what controls the longevity of organic matter in soil. There might not be one answer either, right? I mean, soils are so diverse. There's so many differences, so many different controls and scenarios. But what is clear, and what I think every soil scientist would agree with, is that we need to better understand the primary drivers that are controlling the longevity of the soil organic carbon. You know, some policyholders are already suggesting that we need to throw a lot of effort into increasing the amount of organic carbon in soil as a way to mitigate potential climate change effects. But what we also know is that in some cases you can dump a lot of carbon into soil and it just becomes respired. There's not much net gain. So what we're trying to do is we're trying to develop a new tool to really help us better interrogate the organic carbon in soil, right? Historically, we've had for decades, we can look at radiocarbon dates of that carbon in soil, and that tells us something interesting. It tells us how long that carbon's been there, but it doesn't necessarily give us any history about how long that carbon's been present in a specific molecule. There are literally thousands of organic molecules in just a small handful of soil. And what we want to understand is what are the controls on locking carbon in one molecular type versus having a lot of reorganizations and transfers from one molecular pool to another. So the key thing that's missing in order to do this, and I will preface this all again by reiterating that this is definitely a work in progress, so I don't have the final outcome at this point, but a key tool to really unlocking the data that we need is the ability to make radiocarbon to some degree, but definitely tritium measurements on very small sample sizes. So we believe that tritium measurements can help us understand the sort of hydrolysis and reformation reactions of organic molecules in soil, so that if you can understand, really make fine-scale measurements of the tritium age of organic molecules combined with the radiocarbon age, you might be able to push forward and really get a better, clearer picture of what a recalcitrant organic molecule either looks like or what environment is really preserving it. But because there's so many organic molecules in soil, you can't simply extract all of them and pursue traditional tritium measurement techniques. You would literally have to extract like a dump truck full of soil. And that's just not something that's feasible, nor is it even scientifically valid, given all the heterogeneities and things like that we count in soil. So up on the slide, our task that we're really trying to solve here is that we wanted to improve the tritium measurement sensitivity to help us unlock further investigations of soil organic carbon, which we think have implications for society at large, as well as a great deal of scientific insights. So while we're thinking about this, on one side of campus at Pacific Northwest National Laboratory, we're thinking about this, environmental science sort of stewing over this. On the other side of campus, our physicist friends are doing this. What they were doing is constructing a P&L shallow underground laboratory. And I really want to say it's a laboratory, right? It's not only a measurement facility, but there's a lot more components to it, right? So working in this diagram, basically from the right side to the left side, there are facilities here for purifying materials, machining these materials, taking the purified materials, making detectors, making counting chambers, and then going through and actually performing measurements on them as well. So what our physicist friends were really thinking about is a couple different potential applications, right? They're thinking about contributions to dark matter experiments or probing neutrino masses and things like that in part of their minds. They're also thinking about identification of treaty violations, specifically with nuclear testings and things like that. What they really need for those applications, as we've heard already this morning, is low background materials, right? They have to be able to get very low backgrounds if they are to make their measurement, very fine scale measurements on small sample sizes with not a great deal of activity. So they've leveraged a lot of their work here, really focusing on identifying pure materials. So they'll take here the purest copper that they can find, and then they'll further refine it, right? Trying to improve the purity as much as they possibly can to reduce that background and thereby increase their measurement sensitivity. The nice thing about this lab, the way that the lab's set up, is that once they have that copper, they can apply it for multiple different things, right? So here they're doing some electroforming of copper around a mandrel to make a proportional counter, which might be very relevant, for instance, in environmental applications. But they've also done a great deal of work. They can modify, because of the nature of this lab, they can modify those pure materials. You can see polymers here, copper, a whole number of materials. They can modify those materials into a very wide variety of different components. And then they can apply those components to make counts in this exact lab. So here you see a proportional counter being installed with a sample in it. It really depends on your perspective as to what this is. I mean, one could view this as a device for ensuring that no nuclear treaty has been violated. One could also, if you put your environmental science cap on, view this as a cutting edge measurement, trying to probe soil organic carbon or a whole suite of other compounds. And what we, in the same, I should say the same goes when you start actually performing counting, right? When you actually want to count the carbon or the tritium in these samples and put it in the counter, all the shielding, the veto panels, all of this material is equally suited for enabling a wide range of applications. You know, here we might be counting an environmental sample next to a sample used for very different purposes. So when we got together with our environmental scientists, we learned about what the physicists were doing, what they were constructing with this laboratory. And it quickly became apparent that there could and should be some room for collaboration here, which is what I really want to mention today. However, you know, we felt like the two pieces of the puzzle were present at our institution. We had a tremendous clean room facility in this shallow underground lab that was capable of doing fantastic measurements. We also had a very real and persistent environmental science question. But what wasn't immediately apparent was the bridge to connecting those, right? So luckily for us, we have some internal funding opportunities. And one of those internal funding opportunities was really used to help make that bridge. So first I want to talk about just how close yet how far away apart these two, environmental science and counting capabilities actually were. So think about environmental science, the application. Literally, we want to go into the ecosystem, sample soil, put it in a jar, bring it back to the lab, extract specific compounds out of it, and then perform our tritium and potentially carbon dating on it as well. Now, the shallow underground lab is where we want to do our counting. Keep in mind that the whole lab is a clean room, right? The whole lab is a clean room. And if you're running that lab and you're saying, whoa, what do you want to do? In your mind, when I say I want to bring soil into your lab, you might view that as you want to bring dirt into my clean lab. That's sort of a non-starter. But what we're able to do is really use some internal funding to help make this bridge. So we're able to construct different combustion techniques, different chemical reactions to really convert our targeted sample into, in this case, methane, to perform an analysis, and then really clean that up to the satisfaction of the lab stewards, so that we could then bring our sample into the facility to leverage the incredible measurement sensitivity that they have. So really, this internal funding opportunity helped support the development of this entire capability that really bridged the environmental sample with the cutting edge measurement sensitivity. And as a result of that, I like to put this picture up as kind of a summary. Previously, if you wanted to do all sorts of different types of tritium measurements, you might need multiple milliliters of water in this case derived from your sample. We've been able to reduce that to needing only on the order of 100 to 120 microliters of sample, and still get very relevant tritium measurement capabilities out of it. And what we feel this has done is really help us develop a current capability that really connects environmental samples with high sensitivity counting, right? So, on the slide here on the left, you can see bioenergy feed crops. You can see natural forested ecosystem, agricultural settings, maybe even looking at plant tissues and biomass. We're able to connect that, and on the right, you can see that we can make tritium measurements here from about 0 to 20 keV, and also with some radiocarbon measurements. And this is, I think, only possible due to the collaborative interdisciplinary team that we had working on this process. So our current status here is that we have continued work. That this internal funding brought some people together. The internal funding is gone now, but the people have stayed together, right? And I think that this is really a testament to sort of the interdisciplinary nature of the scientists working here. We've begun in some ways to learn each other's vocabulary, almost to think like each other. And I think bring opportunities to each other and really try to work together to make those opportunities work. We believe that this has opened up some scientific horizons. We're currently looking for some funding to pursue some of our original goals, but we have successfully sort of landed some other opportunities that really built directly upon the infrastructure that we had here. And I say infrastructure, I use that term broadly, using it both the sample handling infrastructure, but also the collaborations, the specific people that are now connecting that we didn't have connecting before. And I want to also say that I think this was all made possible by the really the openness to scientific collaboration at our institution. We're really not siloed in any way, but just taking we just need a catalyst, I think, to begin interacting, and that was demonstrated in this case. And I think there's an overall scientific sort of paradigm or philosophy that working across fields can help strengthen all of our efforts and improve the overall performance of our larger institution. So I think that's all. I realize I'm between you and the break, but I think it looks like we have a couple minutes if there's any questions.
Yes, thank you very much. That was very interesting. I think especially what you said was speaking the same language and maybe having some small project as a catalyst, and then you don't know what can grow out of that in the future.
We've been very surprised. I think some things that none of us would have envisioned a few years ago have actually come to pass because we were talking, right? Thanks to this initial project.
All right. Are there any questions for Jim? Everybody's very quiet.
Uh-oh, sorry.
All right. But can I ask all the speakers to, since none of the slides are uploaded to the agenda, maybe you had problems. Can you just send me the slides or send me a link and I can do that? And ideally do it right away so I can upload them right now before the panel. So we have a 30-minute break now before the panel discussion starts. And you would see all the speakers again and a few additional people. And please remember to submit questions beforehand or
So
during the discussion.
another thing, sorry, Petra, to interrupt. Another thing, I will end the session, the Zoom session, and then restart it. And you'll have to reconnect to Zoom for the panels. So, or if you're using YouTube, that same thing. That way I can save the recordings. And if you go to YouTube, by the way, you can go back already now and look back at the history and replay anything you want. Okay, back to you, Petra, if there's anything.
I was done, so enjoy your break. See you in a half an hour.
Okay, so again, I will end and restart. Thank you.